All right, guys, today we're going to talk about genetics and DNA. So this is chapter 10, lesson 1. The title of the lesson is the genetic code. And on your page in the textbook, it is 386 through 391. So first off, let's start by looking at chromosomes and DNA. We've talked about this a ton, all right? But we've yet to actually get into it, all right, and break down what DNA actually is and what it does. So if we look at page 387, there's an activity there that asked you to sequence the structures in order from largest to smallest. And they give you the snail kite, okay, the actual organism. So the next largest would be the cell, okay? So if you remember, we have 23 pairs or 46 chromosomes in the nucleus of every cell in our body, okay? That has a nucleus. Obviously, red blood cells don't have a nucleus, so they do not contain DNA. But every cell in our body that has a nucleus, okay, has our genetic code inside. Okay, so all 23 pairs of chromosomes fit inside the nucleus of the cell. So let's look at the individual chromosome. Okay, so there's two legs, okay, these are called chromatids, and they're held together in the middle, if you remember, by a centromere. So what a chromosome is, and if you look, it's like pulling a thread on a sock, okay? And here's the thread, and all that chromosome is a gigantic strand of DNA, okay? So we're going to get into this a little bit later. So we've talked about genes before and how genes code for a protein, and it's that protein that gives us our phenotypes or physical traits. So what a gene is just a section of DNA. Let's say from here to here would be a single gene. Okay, we have about 20 to 25,000 genes, okay, that make up the human genome. So this would just be one of 20, 23, 25,000 genes. All right. So if we continue to break down DNA even more, we see these little letters here, AT and CG. These are what are called the nitrogen bases, all right? And we're going to get into this a little bit here, a little deeper here. The arrangement of these base pairs, okay, are called, and they code for what is called a codon, okay? A codon is a section of 3 base pairs, and that makes a particular amino acid, okay? And if you remember, proteins are huge chains of amino acids. So the sequence of these base pairs determine, excuse me, what amino acid gets made, okay, and ultimately what protein gets made, and that protein determines, okay, the particular gene. So hopefully you watched the intro video that I posted yesterday. If you didn't, I would highly recommend watching that before reviewing this PowerPoint. So, like we just talked about, the DNA contains the genetic info, okay, for the cells to make a protein. And again, it's these proteins that create our phenotypes, okay, or physical characteristics. Hair color, eye color, nose shape, height, attached earlobes, hitchhiker's thumb. These are all traits that are physical, all right, and are determined by the protein that is made in our DNA. So again, I've talked about, I just went through the structure of the DNA. Our chromosomes are made mostly of proteins in DNA, and this is found in the nucleus. The actual structure, that picture that we think of when we see DNA, it's a shape, and it's called a double helix. So it's a twisted ladder, okay, and that is the shape of the DNA. So if you think of a ladder, the sides, okay, of the ladder, the rails, are made up of two different structures. One is a deoxyribose, okay, which is a sugar molecule, and a phosphate molecule. Okay, so that's what makes up the sides of the twisted ladder. The base pairs, okay, the nitrogen bases, are the rungs. So if you think of the rungs, that would be where you'd put your hands and your feet on the ladder. So to give you a look at it, and this is over on page 388 and 389. So here's the sides. This would be the rails of the ladder. Okay, so here you see your deoxyribose, okay, and then held together by a phosphate molecule, deoxyribose phosphate. Okay, so this is, like I said, the sides or rails. And then if we look at the rungs of the ladder, okay, we see our nitrogen bases. Okay, so here's adenine, and just by our base pairing rules, I know that this is going to be T, this is going to be thymine. Okay, A always bonds with T, C always bonds with G. Adenine to thymine, guanine to cytosine. And they're held together by a weak hydrogen bond. Okay, so it's just it's a chemical bond that holds these nitrogen bases together. And think of the poles on a magnet. Okay, so if you would try to put, say, A to C, it would push away just like a magnet would push away. And then when you get the polarity correct on a magnet, A would immediately bond with T, adenine to thymine. And the example they gave in the video that I showed, apples and trees, cars and garage. Adenine, thymine, guanine, and cytosine. Okay, those are your base pairing rules. And make sure we know the structures of the DNA. Okay, so this is what I just went over: A to T, okay, and G to C. This single ringed and double ringed. Pyrimidine, not really important for anything that we're going to do. You'll get more into that with biology, but it basically helps you understand how they bond together: a double ring to a single ring, and a double to single. So if we look here, okay, so I mentioned before that a gene is a section of DNA, all right? And it contains the information that makes a specific protein. So again, the order of those base pairs, okay, in the codon codes for a specific amino acid, okay, and proteins are huge chains of amino acids, all right? So again, we've been over this. As far as the genes go, I have a picture here coming up in the PowerPoint that's going to show how on a specific chromosome, that gene is in the exact place on each chromosome. And as we get into the next couple lessons, we're going to talk about dominant and recessive forms of a gene. And we've touched on that before as well. As we know, each individual organism has a unique set of DNA, okay, that is exclusive to them, all right? And as I mentioned at the very beginning, okay, DNA is found in every cell in our body that has a nucleus. Okay, obviously red blood cells don't have a nucleus, so DNA cannot be extracted from a red blood cell. So if we look at genes and chromosomes, so again, here's our chromatid. Okay, this is another chromatid held together by the centromere. And this is just an example, okay, of genes that, something like eye color. I believe it's 7 different genes that all code together with different proteins to determine the pigmentation of the eye. Okay, so this is just an example to show you where genes are on chromosomes. You know, I made the example in class before talking about height. If height was just a single gene that was dominant or recessive, we would either be, you know, 8 feet tall or 2 feet tall, and there'd be nothing in the middle. All right, so height is not controlled by a single gene. It's multiple genes and on multiple chromosomes. But this is just to give you an idea of, to show that specific genes are located on specific spots on individual chromosomes. All right, so if we look at the order of the bases, we're on 389 here. So the gene contains the code, like I've been over several times now. The 3 base pairs together form a codon. Okay. Again, this is a little more advanced than our book is going to talk about. Hey, nothing that we need to go crazy in, but I know some of you are curious, so I bring it up. So this codon, the order, whether it be CCT, AGC, whatever the order of those 3 base pairs are, determines which amino acid gets made. Okay, and these amino acids all link together to make specific proteins. Okay, so again, like I said, the order of the nitrogen bases, okay, along a gene, and this is what forms the genetic code and specifies what type of protein will be produced. Just to give you an idea of how large the ATGC sequence is, okay? If somebody was going to recite the entire sequence on all 46 chromosomes, it would take them 57 years if they did it 24 hours a day, 7 days a week, nonstop. It would take 57 years to go through the entire sequence. So we're talking about a lot of base pairs. So moving on to genes and proteins and how they actually form a trait. So again, like I said, eye color is not a single gene trait, but this is just an example. So here is the specific gene on the chromosome. That's what it would look like as a section of DNA. Okay. The order of amino acids and which amino acids are found in this gene and how they're linked together determines which protein gets made, and ultimately that protein determines our physical trait, whether it may be blue eyes, or hazel eyes, or dark eyes. All right, so this is how those 3 tie together. Here's a look at a single gene, and again, you can see that all these different proteins, and this is just our 3rd pair of chromosomes. Okay. So again, we're talking 20 to 25,000 genes in our human genome. Okay, this is people. Doctors study just diseases, based on genetic information. This is a huge field with a ton of information in it, and it's a lot more complex than we need to get into right now. But I feel like we should have a base understanding of this. All right. So if you remember back when we were going through cell division or mitosis, we always talk about the DNA copies itself. Okay, and we kind of left it at that. But now we're going to take a look at how it actually copies itself. So when we make new cells, obviously that DNA has to be identical. Okay, so the DNA has to get copied. And this is a process called replication. Okay. So if you remember way back in probably mid-September, we talked about the chromosomes unwound, and we just ended up with a ball of chromatin. And that's just a huge strand of DNA, okay, that's not condensed into chromosomes. So what happens is in the nucleus, there's an enzyme called helicase, okay? And the helicase breaks these hydrogen bonds along the base pairs and causes the DNA ladder to split. And if you look over on 390 and 391, and that's what this picture is showing. So the helicase breaks these weak bonds and causes the ladder to split open. So now we have these nitrogen bases that are basically don't have a side. It's just one, the ladder has been ripped in half. Also inside the nucleus of a cell, they're free-floating nitrogen bases. So we have, adenine floating around and thymine and guanine and cytosine, and they're all just kind of floating around in the nucleus. And then when the when the DNA splits, that creates an opening. Oh, excuse me. I have a drink of water here. All right, let's see if that clears it up. So now we have these open ends of the two ladders, and because of the base pairing rules, the nitrogen bases just start attaching to these free-floating base pairs, and probably a little easier for me to show you here on this diagram. So here's our chromosome, it's all unwound. And so the original strand of DNA is here, okay? It is split, and here we have, okay, these are the open base pairs that I was talking about, and now we're just, these free-floating base pairs within the nucleus are just going to come and, okay, and then match up with their base pair rules. There's also deoxyribose and phosphates in the nucleus, and that will form the rails of the DNA. Okay? And then we end up with an identical strand of DNA from the original, and that's how the DNA copies itself for cell division to take place. So again, to go through our nitrogen bases, let me go back, sorry. And very important here, A to T, G to C. All right, so that essentially wraps up lesson 1. Like I said, if you did not view that YouTube video that I posted yesterday, make sure you view that. That is really helpful in understanding what we just talked about today. So tomorrow we're going to get into heredity, and I'll make another video on that. So if you have questions, post them here. Have a good day.